Hi, welcome back to the Praxis Test Prep course. My name is Tasia. I'm a former high school biology teacher, and today I'm going to cover some practice problems for the Praxis exam 5236. These problems are going to cover topics like genetics and evolution. Okay, let's get started. Okay, question 1. If an allele were completely removed from a population, then what would be the effect on the population's genetic diversity? So just a reminder that an allele is going to be any two forms of the gene. So it's kind of one of two or more forms of the gene. So it's an expression of the gene. So the more alleles you have in the population, the more the diversity is going to be increased. And if you remove one completely from that population, then the diversity is going to go down. So A, it says the diversity would be increased. That is incorrect. B, additional information about the population is needed before coming to a conclusion. We know that taking out that allele would decrease diversity. So C, diversity would stay more or less the same. We know that that's not true as well. And so our answer is going to be D, diversity would be reduced. Because if you were to take that allele out of the population, you're taking out a form of that gene. Okay, question 2. An autosomal recessive trait is expressed in 4% of the population in a Hardy-Weinberg equilibrium. So these are all very important keywords that we need to clue in on. What percentage of the population are carriers? So let's remind ourselves what Hardy-Weinberg equilibrium is. It is going to be p squared plus 2pq plus q squared equals 1. So just a reminder that p squared is going to be that homozygous dominant trait. 2pq is going to be our heterozygote or our carrier. So that's what we are asked about. We are asked what population is the carrier. So we're asked what 2pq is. Q squared we are given. So that is the homozygous recessive trait or the autosomal recessive trait. So we know that that's going to be 4%. So if we put 4% in a decimal form, that's going to be 0.04. That is going to be q squared. Now we just need to get p. So p is going to be 1 minus q. That's just a quick way to find those p's or q's. And we know that q squared is 0.04. So we need to take the square root of 0.04 to get q, and we are left with q equaling 0.2. So now let's find p. So p, again, is equal to 1 minus q, which now we know what q is. So 1 minus 0.2, which is going to give us 0.8. So p is 0.8. So now let's figure out what 2pq is. So we know that, okay, so 2 times p, which we just found, which is 0.8, times q, which is 0.2 that we saw up above. So now we multiply 2 times 0.8 times 0.2, and we get 0.32, or 32% of the population, if you make that a percentage. So we're left with C, 32%. Okay, question 3. A parent gamete cell has just undergone meiosis 1, producing 2 daughter cells. One daughter cell is found to contain a diploid number of chromosomes for all chromosomes except for chromosome 18. The daughter cell is missing a copy of chromosome 18. How many copies of chromosome 18 would be expected in the second or other daughter gamete? So let's remind you of what meiosis 1 normally would look like. So let me draw a cell here, and we have 2 copies of each of the chromosome lining up down the center of the cell. Let's say that the bottom copy is going to be chromosome 18. Now in normal meiosis 1, so if we split these 2 cells apart to meiosis 1, again, normal, just so you have a reference point. If we split apart that cell, we are going to have each of those pairs separating. So they're each going to get basically one of those copies. We're not seeing that though in our example. So we see a diploid number in 1 except for chromosome 18. So we have what's called non-disjunction happening in chromosome 18 specifically. So when we have that happening, those pairs don't actually separate. They go into one of the chromosomes, they kind of get stuck together. So what you're seeing here is you're seeing in one of your daughter cells, you are seeing that same X, and then you're seeing that second copy being pulled along with it. you are left with both copies in one of the daughter cells, and in the second daughter cell, none. So they gave us that we have none. So they're asking, okay, well, what's in that other daughter cell? So we have n, which is our normal number, plus 1. So the normal number would be 1. So it's 1 plus 1 is going to be 2. So that's going to give our answer. B is 2. Okay, final question. A geneticist is studying a new syndrome. They believe the trait is X-linked. They are interested in determining the pattern of inheritance. That's going to be key there, which explains how they would determine whether the trait is X-linked. So just a reminder to actually talk about the sex chromosomes. Mom has 2 pairs of X chromosomes and dad has an X and a Y chromosome there. So mom is able to give either one of her X chromosomes, and dad can either give an X or a Y. So when you're looking at sex-linked traits, if you are a woman and you are XX, that you're going to get a double dose of that genetic trait. Normally they're not shown and they're just carriers unless it is a homozygous dominant trait. XY, you really are only given that one X-linked trait, so it will be shown if mom is a carrier of that trait. So let's look at our options here. We have a pedigree can be used to determine the pattern of inheritance. If the trait is sex-linked, the proportion of males and females affected will be different. That is going to be our correct answer because we are seeing that a little bit more men will show the trait than women. But let's look at the other options. So B is a Punnett square can be used to determine the pattern of inheritance by determining whether the trait is dominant or recessive. This is just looking at one specific parents, and we need to see that pattern of inheritance. So that's not our answer. C, a pedigree can be used to determine the pattern of inheritance. If the trait is X-linked, only females would be affected. We are shown here that it is actually more men that are affected than women. And D, a Punnett square can be used to determine the pattern of inheritance by showing the number of offspring that will inherit the trait. Again, it's just that Punnett square, so that's just one copy, and we need to see that pattern of inheritance over many generations, not just one. So our answer is going to be A. I hope this was helpful. If you're looking for more ways to study, head over to study.com to check out our other videos. As a study.com member, you get full access to hundreds of practice problems like the ones I just walked you through, as well as targeted instruction for some topics you might still be struggling with, along with some test-taking strategies to help you maximize your score on test day. Finally, we want to hear from you. Please like and subscribe if today's video was helpful. In the comments down below, please let us know if there's a topic you'd like us to cover next. Good luck and happy studying!